respiration time. What organelle does that involve? Oh, the mitochondria, you say. Well, why don't we just zoom in there and take a look. Okay, welcome to the glycolysis pathway. It's going to start with one glucose. Each little Lego thing is a carbon atom. It's a 6-carbon sugar. And we're going to hike down this pathway. And you can think about it as a pathway, almost like we're going on a hiking trip together. So let's go. Let's go on this trip together. Well, we have to pay a toll right away. Can't go until we pay the toll. And that toll is going to be 1 ATP. Great. So now we can keep going down the pathway. Uh-oh, another toll to be paid. And that's going to cost you another ATP. But at that point, the molecule actually splits into 2 3-carbon molecules. The specific names of the intermediates, not important at all. All right, so in order for this one to pass through, it's going to be connected to the formation of, watch the other side of the card, NADH. That's right, so we just made one of those. But there's a second one right here. So this NAD+, which is kind of like a low energized battery that needs to be charged up, it's going to be charged up because of its interaction with this 3-carbon molecule. So this is like a charged up battery, energized, fully charged phone, something like that. So now we have 2 NADH molecules. Let's keep going. So as this passes through, that is coupled to the formation of 1 ATP. And that's called substrate level phosphorylation, and you'll learn about that in your class at some point. And here comes the next one, substrate level phosphorylation. Yay, we did it again. And here we go, another substrate level phosphorylation event and another substrate level phosphorylation event. So now we have these 2 molecules which are called pyruvate molecules. And I have a little picture here which you probably can't see because it's tiny, but that's okay. But they're 3 carbons and we have 2 of them. So the next step then is going to be to move these things across the outer mitochondrial membrane, past the intermembrane space, through the inner mitochondrial membrane to get into the matrix. And that's what we're going to do next. All right, so here we go. We're going to bring one of these pyruvates through, and they're these protein doors that are embedded in the phospholipid bilayer, and there are different ones, and I've drawn them as different shapes. And so here's the second one, and that one's in. Sweet. Here comes the next one through the first door. I think it's called a porin. It's just a non-specific protein channel, which different things can go through. This one here is a little bit different. I believe it's a symport active transport situation, like protons driving the active transport of this coming through. You can confirm that on your own. Here it is, 2 pyruvate molecules. We're in. In where? The matrix. Nice job. All right, this is a short little path, right? We're on a hiking trip together, or a mountain biking trip, if you want to think of it that way. Short path. It's single file, though. It's a very narrow path, if you think of like single file. So they can't travel together. It's one at a time. Okay, your turn. Oh, no, you, I insist. No, you, I insist. Okay, I'll go. My turn. All right, so the first one is going to go, and that's going to be coupled to the formation of another NADH, those molecules that are like charged up batteries. Yeah, there we go. And then we're also going to attach a coenzyme A to this thing, coenzyme A, which is this entire thing, and we're going to lose a carbon dioxide. So this is attached to that molecule. And I need to have some tape to show that it's attached to it. It'll be removed later, but we need it attached now. So this is called acetyl coenzyme A, and there's one of them. Let's do that again a little bit more efficiently this time. Here we go. Acetyl group is going to be attached. Carbon dioxide's removed. So we have acetyl coenzyme A. We have 2 of them here. These CO2s, by the way, are the same CO2s that come out of your face when you're breathing, when you exhale. All right. So we have our 2 acetyl coenzyme A's. These are going to move to the next section of this, which is going to be called the Krebs cycle, still in the matrix. Here we are. So still in the matrix. Hopefully you can see this. Here comes, this is a single file thing again, kind of like one at a time. We have 2, right, but we can only do one at a time. So let's use the first one first. So what's going to happen here is the acetyl group is going to be removed, and then a 4-carbon oxaloacetate is going to click together with the 2-carbon acetyl group. Boom, That's going to make a 6-carbon molecule called citrate. Sometimes the Krebs cycle is also called the citric acid cycle, and that's because the first intermediate that's made is citrate, the 6-carbon thing. And that will interact with NAD+ to make an NADH, that energized molecule. And then in the process, we remove a carbon in the form of carbon dioxide. That's going to be breathed out of your face just like the other stuff. Continuing along, we'll connect that with another NADH that will shed another carbon dioxide. And now we're here. So, all right, this is going to be connected to the formation of a molecule called GTP. It's like ATP, but with a G, so instead of an adenine, it's a guanine. And then that is going to be connected to the formation of an ATP. So that still counts as substrate-level phosphorylation because we're transferring a phosphate off of a substrate here. A little indirectly, but that's okay. So we're back to 4 carbons. We're going to transfer some energy from this into what's called an FADH2. FADH2, these things are rare indeed. You don't see these every day on our little loop here, so that's one of those. And one more NADH molecule made. There we go, and we're back to our 4-carbon oxaloacetate. That's one spin around the Krebs cycle. We have to do it again, so here it goes. The second acetyl group removed. Our 6-carbon citrate is made. Our NADH is made. Our carbon dioxide is shed. Another NADH is made. Our carbon dioxide is shed. We couple this to making a GTP, which then is directly transferring a phosphate to ATP. Our rare FADH2 is made. Ooh, there it is. And then finally our last NADH, and we've regenerated the 4-carbon oxaloacetate. Now all of these NADHs, and don't forget these, NADHs, and don't forget these, NADHs, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and the FADH2s. All of this stuff is then going to find its way to the electron transport chain in the inner mitochondrial membrane, and that's where this video will go next. Starting from where I'm standing, I'm in the cytoplasm. This is the outer mitochondrial membrane. This is some space between the outer and the inner mitochondrial membrane, and this is where all the action is down here. These are proteins that are integral proteins embedded in the phospholipid bilayer. There are some other proteins that move, right? They are movable. These are not movable. Over here is ATP synthase. Oxygen is this little plastic tray. Protons are little yellow things. Proton, proton, proton. All right, let's see how this works. So our NADH molecule is going to get turned into NAD+. There it goes. And these little washers represent electrons, okay? And so 2 electrons. A proton is going to be moved from low to high concentration to the intermembrane space. The CAR here, this protein is going to get reduced, and it's going to transport those electrons over here to this protein. A second proton will be transported to the intermembrane space. A different CAR, mobile protein, is going to get reduced and transport these electrons over here, and we're going to move another proton to this space. Now, the fate of these electrons is over here. So the oxygen atom plus 2 hydrogens and the electrons will come together to make water. So water was just produced. Meanwhile, these protons here are going to find their way to the ATP synthase, where they're going to go through and it rotates the thing and you're going to make ATP. So I'm just going to show 3 of these. One ATP made, 2. It's supposed to come out the other side. 2 and 3. 3. There we go, I got one. 3. So that's 3 ATP. So we're going to do this again. This time we're going to start with FADH2. So FADH2 starts at a different location when caching in the energy. So it starts here. We are going to turn FADH2 into FAD. These electrons are going to get associated with the CAR. CAR is going to transport these things here. That's going to move our first proton across. Second CAR is going to get reduced, transport the electrons over here. That's going to move our second proton across. These electrons are going to end up in water, H2O, water's made. These two protons are going to come over here to ATP synthase, and they're going to go through the thing and make one ATP, two ATP. And that's the basic idea on how FADH2s are cashed in. You can think of this a little bit like baseball with a pitcher and a catcher, that NADH and FADH2 are like pitchers, and oxygen is the catcher. The only difference is that our pitchers pitch from two different locations, where NADH is going to pitch from like a really far away pitcher's mound, and FADH2 is going to pitch from a closer pitcher's mound. So how is it that the NADHs that are made in the cytoplasm during glycolysis, how do they get across the mitochondrial membranes so that they can get to the matrix and then interact with the electron transport chain that's going to be found embedded here in the inner mitochondrial membrane? And the short answer to that is that they don't. But of course they do, so how do they? And the answer to that involves this: the malate aspartate shuttle, which is a little protein system that moves something else other than NADH back and forth. Here it goes. So here's the NADH that we'd like to get to cross the membrane. And to do that, we're going to get oxaloacetate involved. So oxaloacetate, when it converts to malate, will turn NADH into NAD+. So that seems counterproductive if we're trying to get NADH across, but it'll work. So NAD+ stays over here, but malate travels to the other side. So once it gets to the other side, it's going to switch back to oxaloacetate. And when it does that, so when malate switches to oxaloacetate, watch what happens. NAD+ becomes, you guessed it, NADH. So functionally, we did get NADH across the membrane, but it never actually traveled across the membrane itself. It involved this little shuttle system. So it's across. Oxaloacetate is going to get converted to aspartate here in the matrix, and then it's going to go back through the little shuttle thing to the other side. And here on the other side, it just repeats. So that will get turned into oxaloacetate, and another NADH will show up, and malate will be formed, and that's going to make NAD+, which seems not helpful, but don't worry, because malate's going to bring the energy through for us to the other side. Here's malate. Malate is going to be switched to oxaloacetate, which is connected to NADH formation. Yay, we got it to the other side. This goes back to aspartate, and it goes back and forth, and that's how the shuttle gets NADH across the membrane. But if there's no oxygen, we're not going to do any of this. We have to go somewhere else. So we're going to take a sharp right turn here to this part. All right. So pyruvate, we'll go one at a time here, single file. This one pyruvate is going to shed a carbon, and that's going to be turned into acetaldehyde, which is what's drawn here. Now, it's stuck. We need something here. And what we're going to do is we're going to take the NADHs, I hope you can see this, the NADHs that were made in glycolysis, they're going to come over here to this fermentation pathway, and that's going to allow this to pass and become ethanol. But that's going to flip it back to NAD+. Ethanol is 2 carbons. Here's the structure. And then the second one can go. So here's a carbon dioxide to make acetaldehyde. Here's the other NADH from glycolysis, which as it passes will be converted to ethanol and NAD+. Now where are these going? Well, you can follow them. Here they go. They're going right back over here to, you guessed it, glycolysis.